So the disease that we're going to look at is for these sections, okay, this time we're going to look is AIDS, or called acquired immune deficiency syndrome, or acquired immunodeficiency syndrome. Okay, so AIDS is not the virus. The virus is human immunodeficiency virus. As I say that, guys, don't go and tell everybody HIV virus, because HIV, the V already stands for virus already. So it's not human immunodeficiency virus. Okay, so it's just HIV. Okay, so HIV normally we're going to link it with the first chapter when talk about the virus. Okay, in detail, look at the outer envelope of virus contain two glycoprotein, GP120 and GP41. Okay, so their protein core contain the genetic material and the enzyme protease and reverse transcriptase. Reverse transcriptase use RNA as a template. And we have told you guys before about this. Reverse transcriptase as a template to produce DNA once virus is already inside the host cell. So this diagram, you have to know where is a protein code. Highlight protein code. All viruses, they must have protein codes. So protein code actually is here. The outside one is called as the envelope. So this is protein code. So inside protein code, you have the genetic material. So genetic material for the HIV is RNA. So because they have RNA, we want to produce DNA. So you need to have the reverse transcriptase. Are you clear? From RNA to DNA, so reverse transcriptase. Okay, huh? So HIV is a retrovirus. Retrovirus means that they have an RNA as the genetic material, not DNA. And inside the host cells, this viral RNA can be converted DNA under the process known as reverse transcriptase. This process name is called reverse transcription. Okay, so therefore, this DNA can be incorporated into the human genome. Why we want to incorporate? If you don't incorporate in the human genome, humans RNA, the mRNA process won't involve the HIV DNA. Okay, understand, huh? What this means. Now, inside our host cell, our nucleus, we have DNA. So our DNA, if enlarged, we know that it's double-stranded. It's DNA. So DNA, they can be used under transcription process. We can get the mRNA. So under translation process, we get the polypeptide and then folding. Still remember? Clear? Okay. Now, what we're going to talk about here is if the virus, okay, black, oops, sorry, what happened? Okay, so virus. This virus is the HIV. So HIV actually have their RNA. So RNA is a single-stranded as RNA. So you wouldn't be able to incorporate this into the human's DNA. So in order to do this, so the RNA had to be, we use a reverse transcriptase to make this. Okay, and then we only incorporate it. So incorporate, so for example, this region. Can you see that? When incorporate already, yes, then we can do the transcription. We get the mRNA, polypeptide, and this polypeptide will fall into the viral proteins. Are you clear? Okay, yeah. So the virus infect and destroy the cells of the body immune system, helper T cells, so that the number gradually decrease. When the number of helper T cells are low, unable to defend ourselves, and it causes us to have a variety of opportunity infections. So in fact, HIV won't kill us. The one that kill the HIV patient are those opportunistic infection. Okay, So let's look at the life cycle HIV. You look at life cycle HIV. First, they have this glycoprotein. Okay, so glycoprotein can bind to the cell surface receptor. So CD4 cells, they remember helper T cells. So once it bind, you can see that now fusion take place. The envelope release the viral RNA. So this is RNA. So this RNA using reverse transcriptions to form the DNA, and then using the enzymes, can you see that? We integrate it. So blue color is the host, red color is the viral. Okay, so incorporate, and then during the replication process, they make more and more DNA. So this DNA can assemble together, then form new virus. So this is the life cycle of the HIV. Okay, so take 3 minutes, arrange the following events in the correct order. Okay. So for this part, just you need to know, okay, is a quite simple one. So first, yep, HIV will attach itself to the receptor on the surface of CD4 cells. Write down CD4 cells, also known as the helper T cell. Okay. So next, so the HIV envelope and the CD4 cell membrane now fuse, which allow HIV to enter the CD4 cell. So this one is actually named as receptor. Mediated endocytosis. Okay, so after this, inside the CD4 cell, HIV releases and uses the reverse transcriptase. Okay, they always use the reverse transcriptase to convert the HIV RNA into the HIV DNA. So this DNA now enters the CD4 cell nucleus combined with the cell genetic material, which is host cells. Okay, DNA. So once in the host cells, inside the host cell nucleus, HIV releases integrates to insert viral DNA into the host cells. So very important, they must incorporate first. Okay. And then once integrated into CD4 cells DNA, HIV begins to use the machinery of the CD4 cells to make long chain of HIV protein. So the protein chains are now a building block for more HIV. So new HIV protein and HIV RNA move towards the cell's surface membrane. An example to this immediate non-infectious HIV until newly formed immediate HIV pushed itself out of the host CD4 cells. Then this HIV will release protease which act to break up the long protein chains that form the immediate virus. Smallest HIV protein combined to form matrix HIV. So it releases. Okay. So again, it's very simple, guys. What I say that first, you bind, release, reverse transcription, integration, replication, assembly, release. That's all. Okay. So HIV is a slow virus. After infection, they may not have any symptom. Okay, you won't see any symptom until the years later. Some people who have the virus even appear not to develop any symptom, such as flu-like symptom for several weeks after being infected. So at this stage, the person has HIV positive, tested HIV positive, but no AIDS. AIDS is not one single disease, but it's a collection highlights opportunistic diseases. Okay, associated with immunodeficiency caused by HIV infection. For example, Candida albicans called candidiasis, on pneumocystic girovacin cause the pneumonia. Those actually no problem. For us, we have the immune system actually to get rid of them. But because hepatitis cells no function, lack of functions, so therefore you can see that they actually have the oral thrush and the rare form of pneumonia. So as when immune system collapse further, less effective destroying in finding and destroying cancer, so we have the rare form of the cancer, Carboxy Sarcoma. The cancer internal organ are now most likely cause of death of people with AIDS in developed country. Okay, so like degenerative diseases also like brain and dementia, those are the things. Because why? The immune system fail to detect and kill or destroy the cancer cell. So one, this is Carboxy Sarcoma. So you can see that this is the patient. This is hallmarks. Huh? Patient with HIV, you can see that. Or AIDS, you can see this. Okay, this is candidiasis by okay, in the HIV patients. Okay. So AIDS, as I said, that's method of transmission as pathogen. Method of transmissions, how? So semen and vaginal fluid during sexual intercourse, okay, infected blood or blood product, contaminated syringe, mother to fetus through the placenta, okay, or mother to infants in the breast milks. So site of infection, this one. So normally immune cells, those helper T cells or CD4 cells. Clinical features, no need to know, just for information only. So currently you can see that's the heat maps, okay? In this case, you can see that no data, some country no data, okay, because they don't provide data, but actually you can see that's the higher cases, okay, in Africa, okay, where they are not really or educated in this case. So transmission HIV, as I say, that is spread by intimate human contacts, direct exchange of the body fluid during a sexual intercourse, blood donations, sharing of the needle across the placenta, and breastfeeding. Okay? So initial epidemic is a start in North America and Europe. Is is due to the practice of anal intercourse. As I say, that anal intercourse is not lubricated enough. Mucus lining of rectum is not as thick as the vagina, so less natural lubrication. As a result, easily have lacerations. So the fluid and virus can pass from semen to the blood. And having multiple sex partner, homosexual and heterosexual, allow the virus to spread more widely. Another group of people at high risk of infections are the hemophilia. Because hemophilia, they don't have clotting factor, blood clot. So blood clot issues. Due to what? Due to they don't have the factor. Okay? So means that we gave the blood products. So they were treated with a clotting factor, isolated the blood pool from many donors. So therefore, it will cause this HIV, okay, to be spread. But nowadays, very, very rare already because we screen the blood, okay? So preventing HIV, for the present public health measure, the only way is to stop the spread of the HIV. We don't have the cure so far. People can be educated about the spread of the infections and encouraged to change the behavior so that they protect themselves and others. So when a sexual intercourse, condom, femidams, dentadams are used to reduce the risk of infection during sexual intercourse. Okay, so because this condom, femidam is the condom for female, dentadam is for the oral sex. So they form a barrier between the body fluid, reducing the chance of transmission. Now, we do have this contact tracing. Contact tracing is an important part of control spread HIV. A person who is diagnosed with HIV positive and is willing and able to identify the people who he or she has put at risk of infection, then these people will be offered HIV tests, okay, a sex partner. Okay, so injecting drug user advised to give up their habit, stop sharing needle. So in some moments, the country actually give them the free needle. So again, it create a debate, an argument here. Should we give them free needle and needle, right? Or we call needle exchange schemes. They give us the needle, we give them the new needle because they are drug addict. They don't have the capacity to buy the new needle. So in this case, to stop the HIV, we give them new means that this is what we call the needle exchange scheme. But again, is it a good idea? Because now we have to balance between drug addicts and also the HIV. Okay, so blood collected from blood donor is screened for HIV. And this is a routine nowadays, to kill any viruses. HIV testing is promoted among strongly the people high risk group, for example, male homosexual prostitutes, injecting drug user and their sex partner. If tested positive, they can be given the medical and psychological supports they need. HIV positive women in high income country advised not to breastfeed their baby, okay? But in low and middle income advised to breastfeed, but they need to supply a secure supply of drugs during this period as a protection they give against the disease, okay? So why? Because now HIV, okay, the virus can transmit through the breast milk. So if high income, they can actually get the formulated milk. They have the alternative, but low income, they don't have. Okay, so if they have formulated milk, they lack of clean water. So means that contaminations so outweigh the risk of the transmitting HIV. Are you clear? So these are the methods used to prevent and reduce the transmission of HIV or AIDS. Okay, choose some to remember. Okay, for control and prevention. Okay, prevention and control. However, the spread of HIV/AIDS is difficult to be controlled. Okay, why? Because virus have long latent stage, means that it can be transmitted by people who are HIV positive, but no symptom. We don't know. The virus change surface protein, make it hard for the body immune system to recognize it. So until now, no vaccine. In some low-income countries, not all donated blood is tested. In Malaysia, yes, we tested for HIV, but in some countries, the government are reluctant to introduce HIV testing because of the infringement of the personal freedom. We cannot force people to go for the HIV. Okay, we call this testing. Okay, so treatment. So no cure so far, but we can use a drug therapy to slow down. Okay, so however, the drug very expensive, variety of side effect. So from mild and temporary to permanent ones. Okay. So what is the treatment? Treatment, actually, we use this zidovudine highlight, one of the treatment. So this zidovudine is similar to the thymine. What we do now, because viral enzyme reverse transcriptase will act. Okay, nucleotide. But accidentally, they take zidovudine in, it will stop the replication. So it won't elongate anymore, cannot. So means that it will stop the replication of the viral genetic material. But nowadays, we need to do a combination therapy. So combination therapy, very complicated to follow. The pattern timing you must follow strictly. People who are able to keep such a regime can become susceptible to strain HIV that have resistant to the drug. Okay, so basically, you try to imagine we don't give one type of drug. If one type of drug, if this period resistant, disease stop. Okay, AIDS. So we give multiple different type of drug. So it's very unusual to have all of them resistant. Okay, so some of drugs become resistant, we cannot use, but still have effective on other drugs. Okay, so this means by combination therapy. Okay, so with this, I've done for the HIV and AIDS.